Hey guys, today we're going to talk about chromosomal mutations. So if you remember, a mutation is any change in the DNA sequence whatsoever. So we've already talked about gene mutations, and remember those only affect one base pair on one chromosome, only on one gene, typically only one phenotype. Today we're going to talk about chromosomal mutations. Those happen somewhere along the entire chromosome, and usually more than one gene is affected, therefore more phenotypes are prevalent with these type of disorders. So you're not just changing one protein, you're changing many proteins. So a lot of these chromosome mutations have multiple phenotypes or different factors that are affected. So the first type of chromosomal mutation that we're going to talk about today is a deletion. So here I have the original chromosome sequence of A, B, C, D, E, F, G. So in a deletion, it's exactly what it sounds like. Part of the chromosome has been deleted. So here you can see genes D, E, and F have been omitted. So that happens, and that's going to affect multiple genes. So in your notes, there's going to be different examples of syndromes or diseases or disorders that have a genetic mutation such as deletion, and you'll see the different types in your homework. Okay, the second type is called a duplication. Same thing, duplication, genes are duplicated. So here we have A, B, C, D, E, F, G. Okay, so a duplication has occurred here. The third type of mutation is called a translocation. So if you remember, trans means across. So this is when one portion of a chromosome breaks off and moves across to a non-homologous chromosome. So translocation is when a mutation when one part of the chromosome breaks off and it's going to join another chromosome that's not homologous too. Okay, non-disjunction is one of the most difficult ones, but it's basically when the chromosomes fail to separate during meiosis. So what that does is you're going to get too many chromosomes or too less of chromosomes. So here you have meiosis 1, where your pairs separate. They're meiosis 2, you want those sister chromatids to separate, but they don't. So what happens is this would be the normal situation where you're diploid and you end up haploid. Okay, here the chromosomes fail to separate, so you're going to get one extra chromosome here. So we say 2n plus 1. So we know 2n in humans, diploid is 46. So here you're going to end up with one more, so you're going to be 47. An example of this is Down syndrome, where you're going to have one more chromosome. Okay, if the opposite cell is going to be 2n minus 1, they're going to be lacking a chromosome. If the chromosomes fail to separate into its chromatids, okay, or half the chromosome, you're going to have only 45 chromosomes. So an example of that is Turner syndrome. Happens when the X chromosomes separate, so the female ends up being XO. She doesn't have two copies of the X, because in her mom, non-disjunction occurred. You get one cell that has both copies of the X and one cell that has no copies of the X. So when she gets the egg from the other part of the dad, or the sperm from the dad, that's her other X, and she got no X from her mom. If you have any questions, email me, video chat me. The Zoom code is on the portal. Hope you have a good day. It's all going to be okay. It's all going to be a learning curve. I miss you guys already. Okay, have a good day.